Good morning, everyone. I'll be chairing a session on phenotypic responses. And I'm glad I'm the first one of the session, so because there's a line of heavyweights behind me, and so this way you know that it can only get better from here. These two people are co-authors of my talk and are equally responsible for what I'm going to be talking to you about. So, when organisms actually face a rapid change, a large change in the environment, there are a few options that each individual has. They can move to a different patch, to different habitat. They can adapt or adjust their phenotype, or they can die. So at the population level, populations can migrate, they can adapt, or they can perish. These are some ponds within the national park that are quickly seem to be drying up. So the standing paradigm in evolutionary biology is very population and genetics based, and basically gives us an idea of there's a number of genotypes within populations with some genetic variants that are exposed to some environmental changes and experience gene flow with other populations, so that there's some standing genetic variation accumulating in the population. So whenever there's a transient rapid change to a different environment, it undergoes a bottleneck, living on the standing genetic variation they had, and hopefully the population will be rebuilt from that. However, this same situation of a system of metapopulations in which different populations are exposed to different environmental regimes and are interconnected with gene flow, also selects for adaptive phenotypic plasticity. And plasticity is the property of a single genotype to express different phenotypes under different environmental conditions. And there are classical examples of such plasticity as when different Daphnia face different predators, or Arabidopsis experiences some competitors. And I mean, there's ample evidence for adaptive plasticity all over. So taking plasticity into account actually provides a different complementary model of genetic adaptation, and that's one in which genotypes that have evolved under environmental heterogeneity evolved as plastic genotypes, and therefore during rapid transition to a new environment, most of the genotypes would shift their phenotype through changes in gene expression, and over time, if that environment stabilizes, it could undergo genetic accommodation or the extreme genetic assimilation. It is often thought that plastic genotypes are jack of all trades and masters of none, but that doesn't seem to be the case actually. When there's some evidence that it's actually the more plastic genotypes, the ones that are capable of producing the extreme phenotypes, and not that much as the specialists. And there's some empirical evidence for that that was reviewed recently in Proceedings of the Royal Society. So plastic genotypes can produce rather large phenotypic differences. So there must be a reason why not all traits in all organisms are completely plastic. And that might be that there are inherent costs of maintenance of plasticity. And although that is almost a theoretical guarantee, then it's been kind of elusive empirically to find and measure these costs of plasticity. But there are some evidences for that, as in plants, so that the most plastic genotypes in leaf area actually had a lower residual fitness, just for the fact of being plastic per se. Tom DeWitt also found the same effect in the snails, so there's some evidence that there are costs of plasticity out there. So if we have a plastic lineage, a plastic population that is split, and then the descendant populations or the descendant lineages occupy more stable parts of the environment, genetic assimilation is expected to happen exactly because of cost of plasticity, because over time there will be no longer need for plasticity, and costs will be the main reason for it to disappear. So in collaboration with Vijay Jivani, we tried and implement a simple heuristic model trying to compare the adaptive dynamics of populations composed of plastic organisms or non-plastic organisms. And the model is quite simple. We had clonal organisms, and in the case of non-plastic organisms, the organisms inherited the phenotype of their parents, and it was nuanced by the mutation rate. We adopted a model of mutation based on Fisher's geometric model of the effect size on the phenotype, so that they would express a phenotype that was just their parents and changed by mutation. So, and then their survival would be dependent on how well their phenotype matched the environment. If they'd had a reasonable match, they would survive and then reproduce again with that same rationale. Now, when we have plastic genotypes, the thing changes a little bit, because organisms not only inherit their parents' phenotype, but also their range of plasticity, say how much they can vary their phenotype in response to the environment. And of course, both the genotype and the plasticity are subject to mutation. So in the end, the phenotype that an organism expresses is the inherited phenotype changed by the mutation they have experienced and the response to the environment given the range of plasticity. That will determine the match of the environment of the phenotype to the environment, and it may have to pay a price of plasticity costs. So it's a rather simple model. We run a number of simulations, and I'll be going through this graph slowly. So we had a burn-in of 100 generations in which we just allowed the organisms to evolve under quite modest environmental fluctuations just to build up genetic variation and plasticity. And then after those 100 generations, we let the environment fluctuate widely. So then there's a line in gray with the environment, and that changed according to the standard deviation. And then we had the phenotype of the plastics, as you can see, matched very well the environment in situations of high mean mutational change, mean mutation rate, and low plasticity costs. It was pretty not costly for them to express the right phenotype, whereas the genotype of the plastics was not following the environment as closely. In the case of non-plastic genotypes, the genotypes followed much closer to the environment because they didn't have the ability to change their phenotype, but they ended up going extinct because they couldn't track fast enough environmental changes. Plasticity is quantified here in this shady area in the background, and it continuously went up. And throughout the simulations, we observed that the diversity of genotypes, our proxy for genetic variance within population, was consistently higher for the population consisting of plastic organisms than for the non-plastic organisms. Here's another example in which we had a low mean mutational change rate and low plasticity costs. So at low mutation rate, the non-plastics quickly went extinct, whereas the plastic ones, again, tracked phenotypically very well the environment, and their genotype adopted sort of an intermediate value across all the environmental fluctuations. And of course, the genetic variance for the plastics was much higher than for the non-plastics. We did a thorough exploration of all primary combinations, and that way we come up with this plot. This plot in the upper row, we have all the results for the non-plastic genotypes population, and the lower panels are for the plastic ones. We are showing in the y-axis the standard deviation of the environment, so how widely fluctuating the environment was, and in the x-axis the rate of mutational changes. Each column represents a different value of plasticity costs, and of course, since non-plastics have no plasticity, all the upper panels are the same. Just put there for comparison with the plastics. And then we observe that at low plasticity costs, the plastic genotypes outperform the non-plastic ones across a wide setup of environmental fluctuations. So they're much better at coping with environmental fluctuations. And then, of course, as plasticity costs go high, then the pattern becomes more and more similar, because plasticity is penalized, and actually within the realm where non-plastics do okay, plasticity costs actually hold the population down. Just to show you now that this is one of these pixels is the result of several replicates of several runs. I'm just going to be showing you this transect more in detail. So here we have different panels in which we model different levels of environmental fluctuation. At low environmental fluctuations, both populations of plastics and non-plastic did quite well. And actually, the plastics pay the price of being plastic. Whereas when environmental fluctuations started becoming important, we see that they both suffer important bottlenecks. The non-plastics actually go extinct. And here you can see the actual individual clonal lineages mapped, and you can see the repeated bottlenecks that end up driving them to extinction. And in the case of the plastics, sometimes they recover, sometimes they didn't. In this case, they got lucky. So plasticity is important, and I can't emphasize this enough, that it helps maintaining genetic variation. And it does that for mainly two reasons. One of them is these, they suffer lesser and fewer bottlenecks. So that alone, that would explain part of why they maintain higher diversity. But also, you have to bear in mind that a lot of the variants, genetic variants, that would have not expressed the right phenotype had they not have plasticity, managed to actually sort of phenocopy the right phenotype. So those two properties of plasticity actually ensure a higher genetic variance within populations. We also modeled specific scenarios in which we had a rapid and directional change in the environment. And that would be kind of trying to play around with situations of global change, for instance. And in those cases, even at relatively high mutational rate changes, the phenotype, the non-plastic genotypes would quickly go extinct. The phenotype of the plastics matched the environment pretty well throughout most of the simulations. And what I think is interesting is that the actual genotype of the plastics took quite a long while to actually reach the appropriate value to the environment they were evolving to. So in the sense, the plasticity was buying time for the right genetic variation to accumulate and allow adaptation. When costs of plasticity were high, once they reached the second environment and stabilized, plasticity was lost. And actually, here we can see over the time of the simulation, first of all, plasticity was relatively low or very low. And over the time of the environmental change, it goes up quickly. And when the environment stabilizes again, it goes down again. So we observed genetic assimilation within our model, an emergent property of it. That sort of at some points remind me of what Christian was just been discussing in terms of that apparently some populations that were not well matched to environment were those that had low plasticity, for instance. So I'm just going to try and give you a quick example, an empirical example that I think it could be illustrating this theoretical model. Here in Doñana National Park, we have experienced the case of one of the largest or more important invasive species, is the red swamp crayfish. Crayfish was introduced in Doñana in about 35 years ago and has expanded throughout the park irregularly since then. In these ponds in Doñana, we have some tadpoles living, some native tadpoles. And tadpoles are like candy bars to a lot of predators in the water, both vertebrates and invertebrates. And all sorts of creatures seem to like to eat them. Here we have some Dytiscus larvae, some aquatic beetle larvae, and some anax, some dragonfly larvae. And all of them eat them. These are native predators, and tadpoles respond to the presence of these predators by reducing their activity rate and also changing their morphology, and especially the tail depth and the pigmentation. Now the question was, are these animals capable of responding to a novel environment that has been introduced very rapidly? And would they respond to the invasive crayfish? So we did some experiments with common frogs and tadpoles, in which in a common garden experimentally, we exposed the tadpoles to either clean water, non-lethal presence of a dragonfly. We had a dragonfly caged in the tanks. Dragonflies that were being fed with conspecific tadpoles, then invasive crayfish and crayfish fed on tadpoles. This way, by distinguishing between fed predators and starved predators, we could tell apart the response to predator cues and combined predator cues and alarm cues from conspecifics. And we saw that there was a clear reduction in activity rate and an increase in tail depth and tail coloration in common frog tadpoles in response to the native predators, whereas they did not respond against crayfish, and only a little bit so when the crayfish were actually feeding on tadpoles. The question, of course, is, well, maybe they were just showing the appropriate phenotype against crayfish, but that doesn't seem to be the case, because we took pairs of tadpoles exposed to and unexposed to native predators, just to create pairs of induced and uninduced tadpoles, and we exposed them to real predation to predators. And in both cases, induced tadpoles survived much better. So it looks like they should be responding to the crayfish, and they're not. So therefore, we concluded they probably couldn't detect them. We have recently repeated an experiment with a different species, the spadefoot toad, and again we see the same pattern. They quickly reduce the activity level in response to native predators, whereas they do not respond to crayfish. However, recently we have discovered that they can learn, although they lack innate recognition of the invasive species, they can learn to recognize it as a hazard, as a risk. And we did that by taking tadpoles and exposing them to cues from the crayfish, and having some siblings from the same clutches and conditioning them by exposing them to both the crayfish and cues from attacked conspecifics. So it was a very Pavlovian approach, and doing this twice within 48 hours, that was enough for conditioning tadpoles, so that afterwards, just by treating them with crayfish cues, the conditioned tadpoles reduced considerably their activity rate, which they were not capable of doing that only a few days before. And this actually made a difference in terms of survival, so that conditioned tadpoles survived better and for a longer time in cases of real predation experiments. So they seem not only they seem to be able to learn to recognize them, but they actually seem to make a difference in terms of survival. Now, is there any chance that this is going to evolve? So far, learning and plastic behavioral responses might be buying time again through plasticity for innate recognition to evolve. And I think there's a margin for hope, because there are some other amphibian examples in which they have evolved innate recognition over time, sometimes over long spans of time. We're talking about historical times, and sometimes as quickly as about 100 years. In this case, midwife toads from Mallorca have evolved innate recognition of nitrogen snakes that were induced in historical times. But it's so fine-grained, the way they have adapted to them, that they actually only recognize snakes that are from the same populations that the snakes originated from, and they do not recognize other populations from the same species of snakes taken from other parts of the coast. So they're extremely specific to the actual cues of the snakes that were actually introduced in the island back in the time. But it looks like within a reasonable time frame, amphibians can evolve innate recognition. So plasticity might be buying time for that to happen. And so I would make that argument that plasticity would express the right phenotype and allow them to for genetic variation to occur. So I think that accounting for plasticity expands our view of adaptation in several meaningful ways. One of them is that the environment is as much as a phenotypic inducer as a phenotypic sieve. So it's not only selecting out some genotypes, but it's also inducing adaptive phenotypes. And that's quite important, and because it accelerates the pace of adaptation. Also, mutations do not necessarily need to precede the adaptive phenotypic changes, but may actually follow later. And that also ties up a little bit with the talk we have just heard by Christian, in the sense that phenotypic responses seem to come first, possibly evolution comes later. Plasticity also synchronizes shifts, phenotypic shifts across the entire population in response to environment, because the environment is an inducer. It's kind of a switch, and most of the genotypes will be switching the phenotype synchronically, which is critical in terms of the pace of adaptation, because otherwise we would have to wait until beneficial mutations spread out through the population and sometimes slowed down by drift. Plasticity also allows faster achievement of adaptive phenotypes, and often are capable of jumping over different adaptive peaks, because it's capable of generating highly divergent phenotypes. And lastly, it helps colonizing new environments or withstand rapid environmental changes and preserves genetic variation. So with that, I'd like to acknowledge the support of our group, our funding sources, Plan Nacional and Programa Ramón y Cajal for Roger and myself, and a number of people, Carlos Herrera, Sonia Sultan, P. Medalar, and Jordi Moya, for insightful comments throughout the building of the model. And I'll be happy to take any questions.